Hello everyone, it's my pleasure to introduce to you. Potassium sorbate triggers kidney injury via dysregulated intercellular communication. A study combining network toxicology, machine learning, molecular docking, and single-cell RNA sequencing. In this study, the molecular mechanism of kidney injury caused by potassium sorbate was systematically analyzed by integrating network toxicology, molecular docking, molecular dynamic simulation, machine learning, and single-cell analysis. 1. First comprehensive study to investigate the molecular mechanisms of potassium sorbate-induced kidney injury using an integrated multi-omics approach. 2. Amyloid precursor protein was identified as the central hub gene through a combination of network toxicology and machine learning algorithms. 3. Single-cell RNA sequencing analysis revealed for the first time that APP mediates intercellular crosstalk between endothelial and immune cells via the APP-CD74 and APP-PTGER2 ligand receptor axes. 4. Provides novel insights into the roles of oxidative stress, inflammatory response, and dysregulated cell-cell communication in PS-induced nephrotoxicity. 5. Establishes a powerful methodological framework for the safety assessment of food additives and environmental toxins. Acute kidney injury, AKI, is a clinically important syndrome with high mortality and may progress to chronic kidney disease. Potassium sorbate, PS, is a commonly used and highly safe food preservative, but it is a potential risk factor for AKI, and its nephrotoxic mechanism, especially the mechanism related to AKI onset, has not been clearly elucidated. This study conducted a comprehensive investigation into the molecular mechanisms of PS-induced kidney injury, employing various methods such as network toxicology, molecular docking, molecular dynamic simulation, machine learning, and single-cell analysis. The study results indicate that PS interacts with key targets such as MMP9 and APP, affecting lipid metabolism and atherosclerosis, as well as the age-range signaling pathway. These interactions trigger oxidative stress, inflammatory responses, and metabolic disorders, thereby promoting the occurrence of acute kidney injury. System bioinformatics analysis. PROTOX 3.0 predicted nephrotoxicity, with 374 PS targets, 4,037 kidney injury-related targets, and 238 overlapping potential key targets. PPI network analysis, ENCODE, and degree screening identified 10 core targets for subsequent validation. Molecular docking validation. The 10 pixels per inches intersection targets selected by ENCODE and degree bind tightly with PS, with MMP9 exhibiting the optimal binding energy, negative 5.7 kilocalories per mole. All bindings are mediated by hydrogen bonds, demonstrating good stability. Kinetic simulation of APP and potassium sorbate showed stability via RMSD, RG, SASA, hydrogen bond number, RMSF, and free energy landscape. The PBSA method was used to identify amino acids with significant contributions to small molecule binding in the complex system. In machine learning analysis, we performed batch removal on the samples and used multiple models to predict results. The AUC of each model showed that results possess accuracy. By intersecting the genes selected from multiple models for degree, ENCODE, and RF, the final common gene was determined to be APP. Single-cell analysis. After batch processing, endothelial and immune cells are the main populations. APP interacts with endothelial cells, monocytes, etc. via the APP-CD74, APP-PTGER2 pathways. Strong communication signals exist between endothelial cells, monocytes, and NK cells, suggesting this is a key mechanism of PS-induced kidney injury. In vitro experiments confirmed that PS concentration dependently inhibited HK2 cell viability, IC 5035.66 mm, and migration, with a significant effect at 10 mm, and also upregulated APP protein expression, suggesting its direct nephrotoxicity and its involvement in the renal injury mechanism through APP. In this study, we explored PS-induced kidney injury mechanisms via bioinformatics approaches and in vitro validation, elucidating the molecular basis of PS-related AKI. This innovative work identifies APP as the key mediator, offering a new cell biological perspective, deepening understanding of food additive nephrotoxicity, and providing a new theoretical foundation and research approach for AKI mechanism studies and early warning.